I start, thank you, Sebastian, for putting together this session and putting together a really exciting lineup of speakers. I'm really looking forward to hearing what everyone has to say. For my own part, my aim today is to introduce you to network-based diffusion analysis as a potential means of studying social learning processes within freely interacting animal groups. And so to just provide you an overview of what I'd like to cover today, I'm going to briefly introduce what social learning is and some of the difficulties involved with studying social learning within realistic social contexts. I'm then going to introduce network-based diffusion analysis as a potential means of addressing some of those difficulties. And then I'm going to illustrate the use of network-based diffusion analysis with a case study involving the social transmission of foraging information within honeybee colonies. So first, what is social learning? We can define it as any learning that's facilitated by observing or interacting with another individual. So for example, perhaps a young monkey is trying to figure out how to break into this otherwise impenetrable fruit, and the way it does this is by seeing what the more experienced individuals in its group are doing. Or you might have a fish that ends up learning where a profitable foraging site is by following a knowledgeable leader. And throughout this talk, I'm going to contrast social learning with asocial or individual learning, which we can think of as learning that occurs primarily through direct interaction with the physical environment. And researchers have long been interested in social learning in the sense that it might provide animals a way of obtaining useful, beneficial information while avoiding some of the costs that tend to be associated with exploring the environment, which can be time-intensive and in some cases very dangerous. In addition, social learning also can allow the transmission of novel behaviors through a population. And so researchers have also been interested in how social learning might underpin traditions and other perhaps culture-like phenomena in non-humans. So kind of a conventional approach for studying social learning might look something like this, where we pair a trained demonstrator with a naive observer. But these types of studies tend to happen under fairly artificial conditions, and they tend to be very closely controlled. And these types of studies have been really instrumental in showing that animals can use social learning, and they've been really helpful in being able to tease apart some of the intricacies of the process of information transfer. But the kind of restrictive range of social experiences that are typically allowed in studies such as this are really a far cry from the complex and non-random patterns of interaction and association that we typically see in nature, which means it's unclear to what extent that studies such as this can provide us, or what they tell us really about how social learning operates within natural contexts and whether it even does so. So there's been a lot of interest in trying to develop methods that might let us detect social transmission within a freely interacting group. And one of those approaches is network-based diffusion analysis. So the assumption underlying network-based diffusion analysis is that if social transmission is involved in the spread of some behavioral trait, then that spread should follow a group's social network. So intuitively, we might expect that social transmission would be more likely to occur between two individuals that are closely connected within their social network, and perhaps less likely to occur between individuals that are more weakly connected. So this is the model that underlies NBDA. And so the model is going to, NBDA is going to model the rate at which individuals in a population acquire some behavioral trait of interest. So perhaps we could think of this in terms of the rate at which those monkeys figure out the foraging technique for breaking into that fruit. And so all individuals have some baseline rate of learning. So this represents the influence of asocial learning. So when working on your own, what's the rate at which individuals pick up this foraging technique? Then we have this S parameter that's going to represent the strength of social learning. So how does observing or interacting with experienced individuals, does that increase the rate at which these individuals pick up this technique? And that's going to be scaled by an individual's network connection, which is encompassed by this series of terms. What this does is it sums up all of the connection strengths an individual shares with all of the other individuals within its network. But that Z term is going to take on a value of 0 for any individual that is currently still uninformed. So what that means is we're actually only going to sum up the connections with informed individuals. So if an individual is strongly connected to informed demonstrators, the model predicts that's the individual that's going to be more likely to acquire the trait than an individual that's only weakly connected to them. And then this last term just means that the whole equation is really only concerned with those individuals that still have not acquired the trait. And so researchers using network-based diffusion analysis typically have one or more of these aims or objectives in mind. Primarily, the primary one is to assess the evidence for whether social transmission is involved in the spread of some behavior. Usually it's also of interest to identify what might be the general pathways that information spreads through a group, and instead of just being able to say whether or not social learning is likely to be occurring, it would also be really good if we could get some estimate of the strength of that effect. How important is it? And so with the remainder of my time, I'm going to illustrate how network-based diffusion analysis may be used to address each of these objectives in turn, using a data set recently collected on honeybee foraging. So honeybees are eusocial insects. They live in colonies containing thousands of individuals. And reproduction within honeybees occurs at the level of the colony itself, not at the level of the individual bee. So what that means is every individual in the colony is invested in that colony's success. And in large part, this success can be thought to kind of tie into how well a foraging force, the foragers in the colony, are able to bring in food in the form of nectar and pollen from flowers. But the availability and quality of these floral resources can vary greatly over time and across different spatial regions. So as a result, the honeybee has evolved a series of elaborate communication mechanisms by which individuals can learn about foraging opportunities from their nestmates. And perhaps the most well-known of these communication systems is the waggle dance, which you can see this individual in green and white performing. It's kind of going in a figure-8 pattern. And there's a period, it's kind of hard to see in this video because it's more choppy than it should be, but there's a period where it's going to waggle its abdomen back and forth really vigorously. This is known as the waggle run. And the duration of that waggle run and the orientation of the dancer's body during that run correspond to the distance and direction of a resource from the nest. And all the individuals that are following her can use this information to learn that location for themselves. They can go out and find that resource and begin exploiting it. Honeybees also engage in this behavior known as trophallaxis. This is where one individual will regurgitate nectar and transfer it to another individual. So this one on the right, her proboscis is extended into the mouth of the bee on the left, and she is receiving nectar from the individual. And so trophallaxis plays an important role in learning about taste and scent of flowers, and it can motivate individuals to go out and search for flowers sharing that same scent. And likewise, just simple antennal contact with the body of foragers can allow for the detection of odors that are clinging to those bodies and might serve that similar role of motivating foragers to go out and search for similar food sources. So the role of each of these social information systems has been well studied, but the recent work has cast some doubt over whether the waggle dance, particularly the spatial information contained in the waggle dance, is the main driver or even an important driver of honeybee foraging. These studies have employed a manipulation whereby they make it such that the waggle dance no longer contains any meaningful information, but that bees are still expending time and energy engaging in this behavior. So what we're proposing to do is use network-based diffusion analysis as an alternative means of assessing the importance of the waggle dance in driving bees to food, and to compare its relative importance with these other behaviors, and to do it in unmanipulated colonies. So this was all carried out on the campus of Royal Holloway, University of London. We had 4 colonies in this experiment. We have them all housed in this building here. And for each trial, we trained up 2 groups of foragers to 2 separate feeding sites located about 200 meters or so. And at those foraging sites, they could feed on sugar water provided by these feeders. And we scented these sugar water solutions with essential oils so that bees could learn that this resource smells or tastes a particular way. And for every trial, both feeders received the same scent. So they both received lavender in this sense. All right, so why did we have 2 feeders? We used this setup so that we could create on demand an unemployed group of foragers that are receptive to going and finding a new food source. But before I come to that, we were also interested in another aspect of foraging, which is reactivation, which is when bees are returning after a break in foraging to a resource they already know about in order to see if it remains a profitable resource. So on the morning of the trial, we set up both feeders, but one of them we left empty. And then we recorded interactions occurring within the hive to examine whether those interactions influenced the order in which individuals first returned to the feeder that they had been trained to. So the ones that are returning to the empty feeder don't know until they get there that the feeder is no longer profitable. They think they're returning to a profitable food source that smells the same as this one. So that was one of our foraging contexts. In the same trial, we now have this group of foragers that no longer can exploit this feeder and will be looking for another foraging opportunity. So we then wanted to also examine how those interactions in the hive influence the order in which those individuals are recruited to our new feeder. Well, new to them. They've never been there before. So there's 2 extensions to the basic NBDA framework that this study uses that I think are worth highlighting here. The first is that we're using a NBDA that allows us to include multiple networks simultaneously so that we can actually assess the relative contribution in comparison to the other networks. Which is important because if we think of social transmission, usually we'd assume that there are probably multiple behaviors involved in this process, and the strength of social transmission might vary across those networks. So in particular, for every individual that was visiting the full feeder and was interacting back in the hive with its nestmates, we use those interactions to create networks based on dance following, trophylactic exchanges, and just antennal contact with the bodies of these foragers. So all our networks were directed, and links started at the informed individuals and ended at individuals that had not yet at that point in time discovered the full feeder. The other extension to NVDA we're using in this study is to use dynamic networks. So most applications of NVDA have used a static network that is thought to represent stable long-term relationships. But when the process of social transmission and changes in network topology happen over similar timescales, using those static networks might be misleading. So just to give you a really simple example, let's say individual A knows about the feeder, it goes back to the hive, it starts dancing, individual B follows that dance for follows the dance for 5 waggle runs, and it goes and finds the feeder. Individual A is still dancing, and now individual C starts following dances, and it follows 10 dances before it also then finds the feeder. If we were to use the static network that was aggregated over those time points, then the NBDA is going to assume that individual C should be the first one to arrive at the feeder before B, whereas if we use the dynamic network, we can properly account for the temporal ordering of those interactions. So here are networks updated based on when individuals last left the hive prior to arriving at the feeder of interest. So now we can address those 3 aims that I discussed earlier. So the first being, what is the evidence for social transmission within these contexts? So first, we employed an information theoretic approach where we ran every model that included every possible combination of the 3 networks, as well as a model that only allowed for asocial learning, so no social transmission is permitted. We can obtain the AIC values for those models and use those to calculate our model probabilities, providing the weight of evidence in favor of a given model relative to the others. And when we do this, this is for the reactivation to the feeders to which a bee had been trained, so reactivation to a familiar feeding source. The top-ranked model included an effect of social transmission for both the trophallaxis and antennal contact networks, whereas the model that only allowed for asocial learning received essentially 0 support. If we do that same process for recruitment, again, we see that the model that only allows for asocial learning has very little support. Here, though, the top-ranked model is the one that just includes that dance following network. So these results suggest that we could infer a role for social transmission within these foraging contexts. But we would like to know what are the typical pathways of social transmission. Here we're using a very similar approach. Essentially, we're just taking those model probabilities and we're summing them across all the models that include a network of interest. So for the reactivation set, we see that the trophallaxis and antennatal contact networks are very strongly supported, whereas the dance network essentially is not really playing a role. For recruitment to a novel source, it's just the dance network that receives support, but it is very strongly supported. All right, so we've identified what are likely to be the typical pathways of information flow within these foraging contexts. Now it would be good to know exactly how important are these social transmission effects anyway. So this is involving getting the estimates for this S parameter and the associated confidence intervals. But rather than show you the raw values for S, which are not the most intuitive measures, I'm going to show you values you can obtain using S, which are the estimated proportion of the events that occurred as a result of social transmission. So for reactivation, we had 138 reactivation events, and over half of them involved some amount of antennal contact with a successful forager, whereas a much smaller proportion involved trophallaxis, though the 95% confidence interval does not overlap with 0. For the recruitment context, we had 56 recruitment events, and almost all of them involved dance following in some capacity. It's around 97%. So based on these results, we suggest that there does appear to be evidence of social transmission in these two distinct foraging contexts. For reactivation, it appears that the order in which individuals are reactivated to a familiar site is predicted primarily by these patterns of antennal contact and to a lesser extent by trophallaxis. Likely this is involving the transfer olfactory information, which has been shown to be able to trigger navigation. Interesting because recently it's been proposed that maybe the main role of the waggle dance is not recruitment to new sources, but maybe it's actually to reactivate a foraging site or foraging force to familiar sites. But our results suggest that this doesn't appear to be the case, at least within this cohort of individuals that are foraging on this specific resource. Whereas the spatial information contained within the dance appeared to be very important in terms of the order in which individuals discovered the novel site. So to draw to a close, network-based diffusion analysis provides a means for investigating social learning in freely interacting groups. I've tried to highlight recent extensions of this approach using multiple networks and temporal networks. And finally, I've attempted to illustrate how NBDA might provide us a way to disentangle the relative contributions of different social information mechanisms within honeybee hives. And real quick, just want to thank my co-authors, Will Hoppitt, Ellouise Leadbeater. Will has been instrumental in developing NVDA. If you want to download, if you're interested in the code, there's a free version of the code at that link. It is an older version. It doesn't have all the extensions that I presented today, but we hope to have a copy with those extensions available very soon. And with that, thank you for listening. And if I have any time left, I'll take any questions. Thank you.